There are other examples where the prior history of activity can affect the rules of synaptic plasticity. And I was really fortunate to do my sabbatical in New Zealand with a colleague named Cliff Abraham, and we were trying to think of a term that could capture the general concept that plasticity rules at any given moment depend on what happened before, basically. And we came up with the term metaplasticity, and metaplasticity is now, um, widely used expression. And basically, in most cases, it's capturing the essence of the sliding threshold of the BCM theory.
Yeah. Okay. That makes perfect sense. I didn't know about this theory, by the way. So this is interesting. So, metaplasticity, it's easy to think about that in terms of something like the critical period, right? There's this critical period, which is the brain has a heightened capacity to engage in things like long-term depression or potentiation. And when that window closes, that corresponds to a lessened ability for those specific types of neuroplasticity to actually be triggered.
The statements you've made are correct, except that the metaplasticity piece of it need not be what's controlling the critical period.
Mm.
So the um, the vulnerability to deprivation diminishes with age, and that's, I'm sure, a good thing. And that really doesn't have much to do with metaplasticity, we don't think. We think that there are very likely, another set of mechanisms that open and close this window of plasticity in the brain. And I think a very simple way of thinking about this is that everything we're talking about depends on the input activity and the levels of correlation. And the input activity is under the control of inhibitory mechanisms in the brain. Now, I get a lot of mileage out of just thinking about excitatory synapses, and I am usually reminded that there's more to the brain than excitation. And in fact, I would say the prevailing theory right now for what brings the critical period to a close is the maturation of inhibitory mechanisms in the cortex. I think there's almost a consensus on that point. I think what's less clear at this stage is what is it about maturing inhibition that curtails the plasticity of excitatory synapses. And sort of an easy explanation, which I'm sure is at least partially true, is that the inhibition filters out the patterns of activity. Since all this plasticity is driven by patterns of activity, if you turn down the volume on those things, you'll have less plasticity. So I think that that's considered the most likely explanation for what's ending the critical period.
And, when we talk about treating something like amblyopia, or just reversing some impairment of the visual system, you've told us about this critical period, this window in which you have the opportunity to make those changes. And if you get past that window, if it closes, then it's much harder to correct the problem. So you have to come up with solutions and apply them during this window. But another strategy that you can imagine in principle is you could somehow induce the brain to become more sensitive and more plastic again. Is that feasible? Is that something people think about?
Yeah, it's great. It is. And so, I just put the kibosh on metaplasticity as the basis for the ending of the critical period. But I will say that we could, perhaps one might exploit metaplasticity to enable recovery of vision in the context of amblyopia or any other disorder of brain wiring. And it was with that motivation that a former student of mine, Betsy Quinlan, who's now at University of Wisconsin, did an experiment that I wish I had done, but she did it, and it was a beautiful experiment. And she took animals that were uh, rendered amblyopic, so they long-term monocular deprivation. They reopened the eye, but there was a permanent visual disability through one eye. So modeling untreated amblyopia in a human. And then what she did was she put the animals in complete darkness. So a completely dark room, meaning zero photons, no light at all. And she left the animals in there for about 2 weeks or so. And she asked so you can see the logic here, I suppose, which is to say, okay, the average activity is definitely different if the animals are in the complete darkness. When the animals are returned to light, are we able to sort of now see a recovery of vision? And that's exactly what she saw. So that when the animals were brought out of the dark into the light with both eyes seeing, she got a very nice recovery of vision, so which otherwise would not have been possible. Now, I'll say that her experiments were done in rats. They're not the, gold standard species, at least for relevance to human vision, but the experiments were replicated by others, particularly Don Mitchell and Kevin Duffy at Dalhousie University, using more traditional species with excellent visual system, and they got exactly the same result. So this was really thrilling that, oh my goodness, maybe we can take advantage of metaplasticity to promote recovery. So there are a few details that are important. One is that it did require a long duration of dark exposure. So minimum 10 days in the animal models.
And when was this happening in development? Remind us.
Oh, these are in adults.
Okay, in adults. Okay, so the idea is you have a strong deprivation in adulthood after critical periods have already closed, and now.
No, let me clarify. So the animals were monocular deprived during the critical period.
Okay.
Induce amblyopia that would not spontaneously recover. So it's just like a child that grows up with a unilateral cataract, and then the cataract is removed as a young adult, but there's no recovery. So that's kind of the analogy.
Got it. So something goes wrong early in development, and vision is impaired in adulthood.
Right. And so what Betsy showed originally was that you put the animals in the dark for extended period of time, and when they come back out, now conditions have changed so that visual experience through the amblyopic eye is sufficient to start to drive an increase in responsiveness to that eye. And so this is a very exciting development. Now, I was going to say the caveats here are that the exposure to darkness had to be prolonged. So, as I said, at least 10 days of dark exposure. And it couldn't be interrupted even temporarily by light. It had to literally be a photon-free environment. You couldn't get the same effect by just, for example, putting patches over the eyes, which leave the retinas still quite active and able to respond to luminance changes in the environment. So we were intrigued by this result. And in the meantime, we were doing some experiments using a compound, a drug called tetrodotoxin, or TTX for short. And TTX is isolated from the ovaries of pufferfish, and it's one of the most toxic substances on Earth because it binds tightly to the ion channels that are responsible for nerve impulses. So, there are sushi chefs that train for years to serve pufferfish sushi, which when it's perfect, it gives just a slight numbness to the lips and the tongue, but every so often they don't get it right, and they'll lose somebody because of a TTX toxicity. So, but it's been an extremely useful tool. And I will say that it is not unreasonable to expect its use therapeutically when you consider botulinum toxin is used therapeutically now as well. So you just have to be careful and use the right dose and restrict the.
Are you referring to Botox there?
Yeah, I'm referring to Botox. So in any case, we were doing experiments with TTX anyway, and we saw Betsy's results, and we decided we would try to reproduce what she had seen with an injection of TTX into both eyes. So the difference between TTX and darkness is that TTX literally abolishes all the nerve impulse activity in the ganglion cells, whereas exposure to darkness leaves many of those cells active. So many cells are active in the dark. So it is qualitatively different, certainly a more extreme manipulation. But what we discovered was that in bilateral injections of TTX into the eyes, both sides, single injection, would inactivate the retinas for about 24 to 36 hours, and then they would come back. But when they came back, we saw a really dramatic recovery of vision in the amblyopic eye. And what was nice about this study was that we also saw it behaviorally as well. So we had animals that were showing behaviorally the visual impairment through the amblyopic eye, and the visually guided behavior came back extremely quickly after this treatment.
And is the injection happening during that critical period?
No, this is adults. These are animals that have been, again, rendered amblyopic. They're adults. There's no treatment. There's no hope. They'll forever be blind in one eye.
And then you inject TTX, completely shut off activity in the eyes. And when the drug washes away, when it wears off, you actually can cure the amblyopia?
Yeah. And so the sort of metaphor that I think works is to imagine rebooting your phone. When things start to act wonky, we all know what to do. We shut our phones off and we reboot them, or the computer, same thing. And essentially what we're doing with this bilateral TTX treatment is we're rebooting the visual system. We're saying, okay, you've got yourself in a bad space. You're in a bad place. We're going to reboot, and we're going to shut everything off, for a day, and then we're going to restore activity, and hopefully now it'll come back, and it does.
And this effect, is this only been done in rodents? Has it been done in primates?
It's been done in cats and mice. Cats and mice.
Is anyone going to attempt a clinical trial on humans?
Yeah, let me take it one step farther. So just let's set that aside for a second. I'm going to take you on a bit of a circuitous journey now, but I'm going to get back to this. So here's the journey. So I've told you that people, there are many millions of people in the world that are adults with amblyopia. They live with it. It does limit things that they can do, and it has societal costs. But they're amblyopic. They see with one good eye. However, occasionally, there is, through a disease or an injury, there is the loss of the good eye, of the fellow eye. So now they're in a terrible situation where the eye that they've been using is no longer functioning. So we would expect from first principles that that's it. You're going to be blind. But what is observed more commonly than not in every case, but more commonly than not, is, and this surprised clinicians, is a gradual recovery of vision through the amblyopic eye, sometimes a full recovery. Okay. So just to recap, so we have an adult amblyope. No treatment would work. No traditional treatment would work. But if the fellow eye is removed, destroyed through disease or injury, then over time there's a recovery of vision through the other eye.
So the bad eye gets better by itself and the
Yeah,
good eye goes
yeah,
missing.
exactly. And so that's kind of a head scratcher. Now, the way people would think about this was they'd say, oh, wow, okay, so the good eye is, sort of exerting a constant suppressive effect on the responses to the other eye. That's called interocular suppression. And that only when you remove that interocular suppression do you see anything coming back in the other eye. And if that were the end of the story, then you would say, well, okay, but nobody's going to lose their good eyes to relieve interocular suppression to help recover the amblyopic eye. But the alternate hypothesis is that it's not the perpetual loss of interocular suppression, but rather it's the temporary loss of interocular suppression or metaplasticity that is creating conditions that allow vision through the amblyopic eye to drive a recovery. And so we can test that hypothesis in animals, and we have. And so I told you about bilateral TTX, but the more recent experiments that we've done, and I really should acknowledge Ming-Fai Fong, who was a postdoc in my lab, who was driving a lot of this research, and Kevin Duffy, who's at Dalhousie. The experiment was we rendered animals amblyopic adults. They could only see through one eye, see well through one eye. And then to test our hypothesis, we put TTX into the fellow eye, but only briefly. So we inactivated the fellow eye, but only briefly.
So temporarily inactivate the good eye, and now the bad eye is the only one you can get visual information through. So it's an analogous situation to what you saw in humans, where they lose the good eye and they're left only with the amblyopic eye.
Exactly. Except that the only difference being that we're going to let the good eye come back.
Yeah. So you're losing
like
vision through that eye.
Good eye and putting it back in.
So you lose vision through that eye because of TTX, and that's 24 to 36 hours of deprivation.
Yeah, depends on how many injections we do, but minimal 24 hours. Sometimes we do it multiple injections. But what we've seen is that the responses come barreling back through the amblyopic eye. And we've seen this effect in mice and cats and monkeys. So now we're getting really excited about this because we think that there is a translational potential to this approach.
So, the implication here is that if you injected a human being with amblyopia, you put TTX into their good eye, that this could cure them.
Yes, that's the hope.
And
that would be the hope.
Is anyone going to literally try doing that in a clinical trial?
I think that it's, safety comes first in all these things, and that we've done a lot of experiments that we are able to do to ensure that there's no lasting toxicity of this treatment, that retinas that are treated are pristine, the visual responses through an activated eye come back fully. So we've done a lot of what we can do. But I think that, we're sort of on a go slow program. We really want to understand the efficacy of the treatment and try to learn as much as we can from animal experiments. But at some point, there'll be a strong case to be made for trying it in humans. And injecting an eye with anything comes with certain risks, but it's also a fairly routine procedure these days. So I think the community of neuro-ophthalmologists are probably divided on whether the benefit would outweigh the risk. But most clinicians that treat people with especially deprivation amblyopia are very enthusiastic about this.
So
potentially.
the idea here is that by injecting TTX, a very powerful toxin that shuts off all neural activity, by injecting that into the good eye, you're inducing metaplasticity, and that is enabling the bad eye to become good again in quite a short amount of time.
Yeah.
So, are there any other strategies that might work in a similar fashion? What I have in mind here is, I talked to Gould Dolan on the podcast, and according to results from her lab, they can actually use drugs like psychedelics to, according to them, reopen critical periods of plasticity. And could something like that be another potential avenue that could be used to do this type of thing?
Yes. It, it is possible, at least in principle, right? I mean, every approach you take is going to come with upsides and downsides. And I mean, I'm a big fan of Gould's work. I can say that we also have done some experiments ourselves where we use the drug ketamine and found that dosing animals with ketamine could restore juvenile-like plasticity in the adult visual cortex. So this is conceptually similar to what Gould is doing. And the TTX treatment, the upside is it's very local to the visual system. It's a circumscribed treatment. There are no systemic effects, whereas psychedelics or other treatment are systemic. And so, you just have to. I guess the big thing is that we're sort of plowing fertile ground now. So we think we have learned that using the knowledge of synaptic plasticity can suggest ways to promote adaptive plasticity in the adult brain. And I think there are going to be a lot of different angles of attack on this, but I'm very excited about that we're going to have a breakthrough sometime in the not too distant future on doing this. And I would also say that the concept, the metaplasticity concept, is a very useful way to think about any type of neurotherapeutic approach to loss of function diseases. Whether it's be recovery from a stroke, for example, or other neurodegenerative diseases, is like, can we boost synaptic plasticity in a way to recover function? And so I think it's a very useful concept, a way of thinking about it, framework to think about neurotherapeutics.
Mm-hmm. Yeah, I suppose, to the extent that reopening critical periods, inducing metaplasticity works, in principle, it has, I think kind of what you were getting at there is it has this potential to be an across the board solution to lots of different types of problems in the brain.
Yeah. I think if yes. Bottom line, yes. Once people, it could be tinnitus, for example. I mean, tinnitus is probably a disorder of synaptic transmission, neuronal excitability. Can we use our increasingly sophisticated understanding of synapses to come up with a way to quench tinnitus?
Mm-hmm.
But that's just one example of many that I think, that it's a way of attacking it. It's maybe a fresh perspective on tackling problems.
Mm-hmm. Have there been, well, let me just ask you this. So, two-part question here. One, can you give us some sense of time for how long before people living with amblyopia there might be actual clinical work that's done that they can take advantage of? And two, in the meantime, since there's quite a number of people out there, you mentioned this is pretty common with amblyopia, they are impaired by it. They're probably very motivated to fix it. Is there anything they can do that might, give them some amount of recovery by, playing around with their own visual system and, forcing the bad eye to work harder or something like that?
I'm not an authority on this, so I hesitate a little bit. But we do know that there are effective alternate approaches to patching that work in children that have to do with what's called a dichoptic display. So in other words, instead of eliminating vision in the good eye and forcing vision through the other eye, sort of you try to take advantage of the good eye to help bootstrap the other eye. So they're called dichoptic therapies, and there's a variety of different approaches. And what's really encouraging is that they can yield outcomes in kids that rival the effective patching, even though patching would be considered the sort of medical standard of care right now. These dichoptic procedures are coming online, and they have a lot of advantages. One of which is that kids don't mind playing a video game or watching a movie through a dichoptic display, whereas they do not like to have an eye patched, nor do the parents. So that's children. I think it's still rather unexplored territory to know how well these things would work in adults. We also know that there are, we talk about plasticity in the brain being gated by inhibition, for example. And there have been studies done where they've tried manipulations of modulatory neurotransmitters that are familiar that normally regulate levels of inhibition in the cortex. For example, acetylcholine is an example. It's a neurotransmitter that is released during wakefulness in the cortex and less so during sleep. And it's been shown to promote synaptic plasticity. And so there are certain studies that have been done to try to boost the levels of acetylcholine in the brain. And you can imagine clever ways of, okay, combining that plus some smart visual training exercises. The plasticity is in there. We use it. It's how we learn and remember. And it's a question of finding a way to tap into it. So all hope is not lost. I would simply put it that way. And that's one thing we've really shown in the animal studies is that we can do it. Reducing it to practice is challenging, but we can do it.